We'll look at the various organs, how these organs work, how the immune system develops, the various cells, and furthermore, we will also look into how this whole process works based upon via some chemicals by which these cells interact with each other. Okay. So, we will look at the main organs, the central and the peripheral lymphoid organs. Then we will look at the cells of the immune system. There is a very important concept regarding the HLA or the major histocompatibility complex, and finally we will look at the cytokines. So, moving on, the first things first, what do we actually understand by the immune system? I think we have already learned a lot about this system, that system, but what actually is the immune system? The immune system is designed to protect the human body from various infective organisms. And this immune system has got two parts. The first part is called as the innate immune system. Innate means that is the intrinsic part of the body. So, whatever may be the environment, whatever may be the organisms, the body has certain defense mechanisms which helps us to prevent the infection. Those are called as the innate immune system. Along with that, there is another part of the system which adapts according to the environment in which the human being grows and develops. That is called as the adaptive immune system. So, if we look at the immune system as a whole, we have certain organs. First, we have the mucous membrane and the skin, perhaps the largest contributor of the innate immune system. The skin and the mucous membrane forms the barrier which does not allow the infective pathogens to enter into the body. Next, if we look at them, all throughout the body, there are some system, it's called the lymphatic system, and comprises of some glands which are called as the lymph nodes, and through them there is a ducting system in which lymph flows. Furthermore, there are specific organs like spleen, thymus, and some of the specific lymphatic nodules in the body. These all comprise the immune system. Okay. Now, if we have to divide these organ systems, we can divide them into two types. One is the central organs and the peripheral organs. The central organs or the primary organs, they are the primary areas. It would include, what is this organ? Where is it located? Yes. So, this is the thymus and this is the site of proliferation of the T cells. I will explain what T cells and B cells are later on. And the next most important part is the bone marrow. Okay. So, what is the role of the bone marrow? As you already know, bone marrow is the site of the hematopoiesis. So, almost all cells that originate, they originate from the bone marrow. Some of the cells will migrate to thymus and form T cells, and some will remain in the bone marrow and later on distribute throughout the body. We call them as the B cells. Now, when we look at the bone marrow, what do we have in the bone marrow? We have the hematopoietic stem cells, and from the hematopoietic stem cells there is a different lineage. One is the myeloid lineage and one is the lymphoid lineage. The myeloid lineage gives rise to what types of cells? Neutrophils, eosinophils, basophils. The other wing will give rise to megakaryocytes, ultimately platelets, and red blood cells. The lymphoid progenitor cells will ultimately divide to form many number of the cells. The most important would be the T cell and B cell. Some other cells would include the natural killer cells. Again, I will come to those later on. Okay. So, the lymphoblast will give rise to T or B lymphocyte based on where it resides and matures, and natural killer cells. So, these natural killer cells, they are the part of the innate immune system, almost similar to the granulocytes, the eosinophils, basophils, which are also the part of the innate immune system. However, the B cells and the T cells, they are the part of the adaptive immune system. So, what I want to highlight is that even though when we first try to understand immune system and divide into innate and adaptive immune system, they are interlinked. They are not separated at all. Okay. Now, so the primary organs would be the thymus and the bone marrow. And if you look at the secondary organs, there would be, now what is the most common? There are groups of some glands all over the body. What are those called as? Those are the lymph nodes. So what are the predominant lymph nodes? Any examples? Inguinal lymph nodes, axillary lymph node, mesenteric lymph node. Okay. Next would be a very important spleen. And last but not the least, there is a specific organ system that is called as, any idea what this is? No. This is called as the mucosa associated lymphatic tissue. Okay. Based on the organ system, we can again divide them and classify them separately. But most importantly, they are associated with the mucosa. Okay. So that was the very simple classification. Now let's move on. Now, during the embryonic development, the first cells that are seen in the yolk sac, and during the fetal development, the progenitor cells comes from the liver. Slowly, they will move to the bone marrow. After the child is born, the cells from this bone marrow will move into the blood. Some of the cells will move to the thymus, mature over there, and then come into circulation. Those are called as the T cells. Some cells from the bone marrow will mature, go to the lymph nodes, and in those lymphatic organs, they will mature. Those are called as the B cells. The B word comes from bursa or equivalent. In birds, there is bursa of fabricus. You can remember it from the bone marrow. And throughout the circulation, all those cells circulate within the blood and the lymphatics via the channels where we have the spleen and the lymph nodes itself. Okay. So this is the normal circulation of the all immune cells. Now, this is the structure of the, let us take one organ one by one. First is thymus. Now, if you look at the anatomy, I think you can easily correlate. The thymus has a medulla, the capsule, and the important artery, vein, and the lymphatic nodes. Now, what actually happens in the thymus is very, very important. What happens over there, if you look over here, this cross-sectional anatomy, in the cortex and the medulla, we have the blood vessels and we have something called as macrophage and dendritic cells. Now, these macrophages and dendritic cells play a very important role in the development of the T cells. How? Let us look in this picture. Suppose there is a particular pathogen or a bacteria that has entered into the body. From there, that pathogen has been broken down. So, when this pathogen is broken down, what happens? It liberates some proteins which we call them as antigens. Now, these antigens will be taken up from the blood or the lymphatics and it will move into the thymus, where the macrophage will engulf those antigens. And the macrophage will then signal via many receptors. One of the very important receptors is called as the T cell receptors. And within the T cells, we will have T helper cells and the cytotoxic cells. Based on the antigen presentation, these cells then will lead to activation of the helper T cells. When the helper T cells activates, we have a two-pronged outcome. The first is the humoral response. This helper T cell will then stimulate a B cell so as to make the D cell sensitive, and eventually an antibody specific to that antigen will be produced. Okay. Now, what is the other wing? That is the cell mediated response. The cell mediated response will happen with the Th1 cell, that means the cytotoxic cells. These cytotoxic cells will liberate many types of cytokines. Cytokines are basically chemical mediators. These chemical mediators will have two effects. The first effect is that cytotoxic cells will degrade the human cells and sometimes even the macrophage itself. so as to finish the organism in total. Have you understood this? This is all said and done very easy. But let us delve slightly deeper and I will explain even more. The problem with this is that, just look at this something. When this cell is activated, the cytotoxic cell is actually inducing apoptosis or degranulation of the normal human cell. In this case, it is an infected cell. But if it is unregulated, what can happen? It can attack the normal cells also. So there is some kind of regulations. So what is the regulation? Just try to concentrate over here. In the cortical epithelial cells, there is a molecule called as the MHC or major histocompatibility complex. This will bind with the T cells and there is a specific ligand called the CD4 cells. The first thing that happens is called as the positive selections. What is positive selection? That means all T cells which can recognize MHC pattern, they will survive. Rest all T cells will die off. Okay? Why? Because MHC antigen is present in almost all human cells. Understood? So if it is restricted to all human cells, it cannot damage any other cells. That is the first thing, that is a positive selection. The next thing that happens is what we call as the negative selection. Why negative selection is important? So that any cell which is a self cell or the human cell is not damaged. And if any human cell antigen is expressed by the T cell, it will be killed. So that whatever T cell that remain in the thymus will be able to bind with MHC molecule only. Understood? Because of this positive and subsequent negative selection, these T cells would not generally attack any human cell until and unless that cell is infected cell or that is a malignant cell. Understood? Okay. Now similarly, let us look at the lymph nodes. I think you are pretty much aware with the anatomy of the lymph nodes. What is the core anatomy? We have a cortex and the medulla. And over here you can see in the cortex there is something called as the paracortical region. So this paracortical region have T cells in majority, whereas in the cortex we have the B cell in majority. So even though we say that B cells originate from lymph nodes, lymph node will also have T cells. Now what is the role of this B cell? Now this is a picture showing the steady lymphatic flow. That means in the uninfected scenario, the lymph nodes are flowing normally in the lymphatic ducts along with the vessels. But suppose the person gets infected with a virus. So this virus will be carried to the lymph nodes either through the vessels or the lymphatic ducts. What it will trigger? It will trigger both the T cells and the B cells. Now the T cells will enable the B cell to form antibodies. How it does? First, the dendritic cells will recognize the antigen and then it will present the antigen to the T cells. It is called as an antigen presenting cell. Thereafter, this happens. This is the normal B cell and this is a plasma cell. What does the plasma cell do? It produces antibody. So next what happens is this B cell turns into a plasma cell. Let us look how. The first, the antigen is presented through the antigen presenting cell via the T cell into the B cell. Now the B cell, when it is exposed with the antigen, it will bind with the antigen and it will form a specific antibody. Thereafter, that specific B cell will divide to form what we call as the clonal expansion. That means that B cell will divide many times to form a clone of B cells, producing antibody against that specific antigen. Now, in acute infections, we have seen there is a rise of IgM and thereafter there is IgG. So, there is a shifting of the antibody that is produced. Similarly, there is also shifting of the plasma cells which produce antibody. Some of the plasma cells will then mature further to form what we call as the memory cells. These memory cells will become dormant, but when the infection happens again, it will start producing the antibody at a higher volume and at a faster rate. That is the difference between the memory cell and the normal plasma cell. So, this is what happens. The pro B cell that is not exposed to the antigen, when it gets exposed to the antigen, start producing the ligand for the antibody production. This B cell then will mature to form plasma cell which will start producing antibody. Understood? So, now till now we have covered very two important aspects. One is the cytotoxic or the cell mediated part and the humoral part. Let us look at another organ that is called as the spleen. So, what if you look at the anatomy of the spleen has two parts. What are the two parts? Red pulp and the white pulp. Now, two different mechanism goes over there. The first is the red pulp. Can you say what is the function of the red pulp? Why it is a red pulp? And why is the other one white pulp? So, in the red pulp, the afferent arterioles will have very small gaps. And through these small gaps, all the red blood corpuscles and the platelets, they are forced to move through these gaps, sieve-like structures. Now, whenever an RBC is infected, damaged, or say an old RBC, the pliability of the RBC decreases. And when the pliability decreases, it cannot come out through these small sieve-like structures and then will be eventually destroyed. So, what the spleen does is it actually gets rid of the degenerated, infected or abnormal red blood corpuscles and platelets. That is the function of the red pulp. Now, keep in mind, there are some bacteria, they have polysaccharide capsules around them. And these polysaccharide capsules are firstly filtered over here. This is the reason why in patients who are asplenic, either because of ম্যাডাম মাথা নিচু করে দেখা যাচ্ছে, আর যদি এতই ইম্পর্টেন্ট ডিসকাশন থাকে প্লিজ বাইরে গিয়ে ডিসকাশন করুন। এতে হাসার কিছু নেই, সব সব জায়গায় একটা ডেকোরাম মেনটেন করতে হয়। যেদিন টেবিলের এই পারে আসবে সেদিন বুঝতে পারবে এখানে বসে বসে পেছনে গল্প ইয়ার্কি হাসি আড্ডা সব চলছে। ক্লাসে আসার কোনো প্রয়োজন নেই, অ্যাটেন্ডেন্স দাও চলে যাও, হোস্টেলে গিয়ে বসে থাকো। Moving on from the red pulp, we move on to the white pulp. Now, the white pulp structure is almost similar to that of the lymph node. If we compare with that of the spleen and the lymph node, we can see it is almost similar. And further similarity can be assessed by presence of both B cells and T cells in both of them. Okay. So, the spleen has dual function over there. As I was telling, in patients who suffer from asplenia, either iatrogenic, that means post-surgical, or because of any defect, the spleen may not function well. So, in these patients, these organisms which have polysaccharide capsules around them, they can cause severe infections. And these types of infections are also called as overwhelming post-splenectomy infections, or OPSI in short. Okay. Moving on to the last part of the secondary lymphatic organisms, it was said that we call it as mucosa associated lymphatic tissue. Based on the organ, they are again named as either nasal associated lymphatic tissues. What would be those? All the lymphatic tissue around mouth and nose. It could be the tonsils, the adenoids, and the Waldeyer's ring. In the lung bed, we call it as the bronchioalveolar associated lymphatic tissue. Similarly, in the gut, we call it gut associated lymphatic tissue, and in case of the genital tract, it is the commonly inguinal lymph nodes. Okay. So, whenever any infections happen, if it happens through these routes, the portal of entry, either nose or mouth, or say lungs, or the gut, or the genitourinary organ, then these lymph nodes will be affected. Now, let us look how this tissue helps in preventing the infection. Suppose we take the lymph node in the gut. What is this? This is a cross section of the intestine. These are the intestinal cells. I think you can observe the microvilli present over there. In this cut section, you can see there are some lymphatic tissue in this. What is this called? This is called as the lamina propria. Similarly, in the submucosa, there is another lymphatic tissue. So, there are two types of lymphatic tissue when it is associated with mucosa associated lymph node. One is in the lamina propria and the other one will be in the submucosa. In some areas, for example, the appendix, the submucosal lymph node are more aggregated. Okay. Nevertheless, throughout the intestine, both these tissue are present everywhere. Okay. Furthermore, throughout the intestinal lining, along with the intestinal cells, we have a particular cell called as the M cells. Now, what is the role of these M cells? These M cells are present within the intestinal cells. And suppose there is a microbe that has entered into the gut lumen. What this M cell will do, it will detect the microbe, then through the many microvilli, it will engulf the microbe and it will try to destroy it. If it is able to destroy it, what it will do? It will trigger the B cells and the T cells and also recruit macrophages. Thereafter, it may trigger the lymphoid tissue either in the lamina propria, and if antibodies are to be produced, it will trigger the plasma cells in the lymphatic tissue present in the submucosa. Now, question is, what type of antibody is secreted from over here? IgA, that is the secretory IgA. Now, please remember, these plasma cells will trigger to form IgA normally present in the blood in a monomeric form. After it gets the secretory protein, it will form the secretory IgA, that is the dimeric form, and will be secreted into this submucosa in the intestinal lining, thereby protecting, or this antibody can also trigger macrophage activation so that the organism can be degraded. Okay. Similar to the mucosa, another big organ is the skin. Inside the skin, we have specific cells. Those are called as what cells are present over here in the skin? What is this cell called? Dendritic cells. These dendritic cells are present on the skin and they can detect various pathogens based on the pathogen activated molecular pattern or the PAMs. The dendritic cells are not able to degrade the pathogen. That means it cannot kill the pathogen. Like macrophage and other cytotoxic cells, the dendritic cells cannot kill the pathogen. But what it can do is, it can present the antigen. Okay. Now, why this is important? If you remember, some of the vaccines are given in the subcutaneous route. and those vaccines are even more efficient. Why? Because whenever we are giving in the subcutaneous route, whatever antigen that is given, those are processed by the dendritic cells, and then the activation based on those antigen happens. Understood? Okay. Now let us look at the cells of the immune system. Till now we have discussed about the various organs. Now look at the cells. Before discussing about the cells, you must understand this thing. This is called as the CD factor or clustered differential factor. These are proteins that are present on all lymphatic cells, and these proteins are different for different sets of cells. And by these CD markers, we can differentiate various cells. Now, if we look at the lymphoid cells as an overhaul, it comprises about 20 to 40% of the total WBCs, and their presence is varied. For example, in the bone marrow, you can see most of them are B cells, very few T cells. In the thymus, it is almost T cells only, whereas in the lymph node, T cells are present about 70 to 80%. Rest are B cells, so majority is T cells. Okay. Now let us classify the lymphocytes. Lymphocytes can be classified into three main types: the T cells, the B cells, and the natural killer cells. What is the basis of this classification? Natural killer cells are cytotoxic cells, but they do not have an adaptive capacity. That is the part of the innate immune system. T cells are also cytotoxic, and some are other helper cells, but predominantly it is cytotoxic cells. But they are dependent on the antigen. That means they are part of the adaptive cells. B cells, predominant role is production of the antibodies based on the various antigens. This is based on the function. Based on the morphology, it can be classified into naive B cells and lymphoblast. Naive means new, rookie. That means somebody who is not been exposed. So naive lymphoblast means a lymphoblast that has not been exposed to any antigen of the microorganisms. Okay, these are also called as the resting bees. They are small in shape and they have a very short lifespan. Whereas the lymphoblast, when the naive B cells get stimulated, both the antigen and along with various cytokines, it forms lymphoblast. The lymphoblast will then turn into two types of cells. One is called as the effector cells. What the effector cells does? It will produce the effect. If it is a B cell, it will produce antibody. If it is a T cell, it will produce cytotoxicity. And the other it can form is a memory cell. That is the dormant form. So when the infection occurs again, it can start the immune reaction more rapidly. Understood? This is based on the morphology. So if we have to differentiate, we have the naive B cell, effector cells, and memory cells. So naive B cell is present in where? Secondary lymph node. Why? Because that is the area where from the portal of entries the organism can get exposed. Next, it lies in the dormant phase because it is not active. They are small in shape and they have a short lifespan. When it gets triggered, what will happen? It will form effector cells. Where will the effector cells be present? At the site of the infection. Okay, where the microorganism is multiplying rapidly, because that is where the action is required. Furthermore, now they have moved from G0 to active life cycle so as to produce various enzymes or immunoglobulin, etc. Hence, the shape becomes larger. However, the lifespan still remains very short. And what it does, it will eliminate the pathogen. Memory cells can be found both in the glands and both in the peripheral tissue. Why? Because the next time the infection occurs, it can trigger more rapidly. It also lies in the G0 phase and the shape is small like that of the naive cell, but their lifespan is very long. Now let us look at these cells one by one. First is the T lymphocytes. T lymphocytes is about 70 to 80% and they do not have villi like the B cells. Villi means these are projections. However, the T cells will have a certain type of specialized surface receptors called as the T cell receptors. Now, this T cell receptor, if you look over here, there is a particular protein called CD3 that is present within the cell. Two more proteins are present. One is the alpha. If you look over here, there is a delta and a gamma. Some T cells will have an alpha and beta. So commonly we call them as the CD4 or the CD8 cells. But some cells may have a gamma and delta. I'll talk about it later on. However, this is the main T cell receptor, a 4-molecule receptor. This T cell recognition is very, very important. If this T cell recognition doesn't happen, it will not be triggered. Okay. So this is the T cell receptor in the complex. Now look at this picture and I'll explain you. Previously I have already told you about the positive selection and negative selection that happens in the thymus. Now let us look at the role. From the bone marrow, the hematopoietic precursor cell moves to the thymus. There it becomes a thymic precursor cell. Now this cell is called as a double negative. That means it neither has a CD4 marker or a CD8 marker. First the CD3 gets attached. Then most of them along with the CD3 will then get both CD4 and CD8 in the T cell receptor. Most of them. But some of them will have the gamma and delta T cell receptor. This gamma delta T cell receptor will form a different type of T cell called gamma delta T cell. Rest all which have the alpha and the beta will have both CD4 or CD8 type. Now, next happens what is called as a positive selection. That means this T cell receptor recognizes the MHC molecule. If it fails to recognize the MHC molecule, the outcome may be, most of the time will happen, is the apoptosis of the T cells. However, some will lose those markers and will form the natural killer cells or T regulator cells. Understood? After the T cell receptor recognizes the MHC, then it will differentiate when it comes into the thymic. All these things happen in the cortex. When it moves into the medulla, it will recognize either CD8 or CD4. The CD8 cell will come out and form what we call as a cytotoxic T cell, whereas CD4 will be the helper T cell. Have you understood? So in the peripheral circulation, we will find either CD8 cell or CD4 cell or gamma delta T cell or the natural killer T cell. Now is it clear? Okay. There is another subset I have already told you, the helper 17 cell. This is the third type of cell. It produces many cytokines. Its role is still not very well defined, but however, in autoimmune and inflammatory diseases, the role have been under study. Okay. Next, we come to the B cells. Now, what is the role of the B cells? I have already told you, it is the production of the immunoglobulin. Maturation occurs with the B cell receptor. Like T cell receptor, it will also have a B cell receptor. Only difference is, initially we can find a surrogate light chain which attaches to the various antibodies, and when specific antibody forms, the main chain undergoes changes. Initially it will be immunoglobulin IgM or IgG, later on IgG. Okay. So this is the basic difference between B cell and T cell. Many a times in your examination, this question is being asked, what are the differences between B cell and T cell? And I think I have discussed almost all of the points. Okay. Now let us look at next. It is the natural killer cell. Why it is called natural killer cell? Because it is a part of the innate immunity. And what it does? Kills what? Own cells. Now if these cells are allowed to act without any specific orientation, what will happen? It will destroy most of the human tissue. But that doesn't happen. There lies the question of something called as this molecule called as MHC molecule. So most of the human cells will show this MHC molecule. Now suppose this is a normal cell, the killer cell comes and it recognizes the MHC molecule. So the cell is functioning normally, it will not damage the cell. Now let us suppose this is a respiratory cell and an influenza virus has entered inside the cell. What the virus will do? It will interfere in the normal protein synthesis of the cell, and among the normal protein synthesis, this MHC antigen will not be produced, or even if it is produced, may be altered. So now the natural killer cell will come and it will fail to recognize the MHC pattern on this infected cell, and then it will lead to degranulation of this cell or killing of the cell. This is one of the way, especially in case of viral infections and in malignancies, these cells are destroyed. Okay. Now let us come and look at the cells which are commonly found in the blood. First is monocyte. What is a monocyte? All are white blood cells, but what is a monocyte? So this is the largest cell present in the blood, and this monocyte, when it moves into the tissue, we call it as macrophages. So what is the role of macrophage? Most important is phagocytosis, right? So when it happens in the tissue, we call them macrophage. When it happens in the blood, we call them as monocytes. Now the macrophages, as if you look at the shape, they will have pseudopodias through which it will form phagosome, and the phagosome, the lysosome of this macrophage will lyse to form phagolysosome, and it will kill the organism by forming reactive oxygen species or reactive nitrogen species and various enzymes. Overall, in the body, we have many types of macrophages based on the tissues. For example, in the liver, we call them as the Kupffer cells. In the kidney, these are the mesangial cells. Lungs, these are the alveolar macrophages, etc. and etc. As we have already said, the primary role of macrophage is phagocytosis. Next role is, if it cannot phagocytose or even after phagocytosis, it will act like an antigen presenting cell. It will process the antigen and it will present the antigen. So after the antigen presentation, it can do what we call as recruitment of the B cells, T cells, cytolytic effect. Furthermore, these macrophage can turn itself into activated macrophage. What is an activated macrophage? There is enhanced phagocytosis and local tissue destruction. It can happen either through humoral activation. Suppose the antibody is present, that antibody is detecting an antigen, or the complement has detected the pathogen, it can trigger the macrophage to form activated macrophage. It has enhanced cytolytic effect. Or other pathways like interferons. Interferons are cytokines. When the macrophages are exposed to interferons, it can turn itself into activated macrophages. Similarly, other cytokines like interleukin 4 and CD cluster differentiating stimulation can also lead to activated macrophage. Why this is important? When you will be leading about, uh, there are two types of infections, inflammation you have read. Granulation, right. So whenever there is a pathogenesis going on, where the macrophages are unable to contain the infection, what it will do? It will start recruiting the B cells, the T cells, release the cytokines, and itself will turn into activated macrophages. So many macrophages will coalesce to form giant cells. Have you understood? So this is where the activation occurs. And we have already said various enzymes, and these are along with the cytokines are produced. Dendritic cells present in the commonly in the skin act as antigen presenting cell. Among them, there is a specialized type of dendritic cell called as the follicular dendritic cells, which are present in the lymph nodes. They help in maturation of the B cells to form specific antibody. Other than that, we have neutrophils. What is the role of neutrophil? Eosinophil, predominantly it is directed against parasitic infections. Similarly, we have mast cells, basophils and mast cells. However, mast cells numbers are more limited and is more consistently associated with the allergies and inflammatory hypersensitivity reactions. Okay, so these were the cells of the immune system. Now, when discussing about the natural killer cell, I have told you about major histocompatibility complex. So, very briefly, what is the major histocompatibility complex? It is a complex of group of genes, and all cells will express them. This terminology, major histocompatibility complex, was observed and named in animals, that is in mice. What we actually have in human beings is called as the human leukocytic antigen, or HLA. Now, these are very important not only to understand how the immune system works, but also remember, during transplantation, one organ is transplanted to another patient. So, there will also be immune consequences. Those consequences are determined predominantly by the HLA. Okay. So, these are the genes where we have class 1, class 2, class 2 and class 3. The specific gene locus will include HLA, ABC and these. They are present on the short arm of the 6th chromosome. Okay. MHC class molecule have two types, class 1 and class 2. The difference is based on this beta 2 part. If we look at the class 1, this beta 2 is not bound to the cytoplasm, whereas in class 2 it is bound to the cytoplasm. Okay. The second important difference is class 1, the alpha 3 part binds with CD8, whereas in class 2 the beta 2 binds with CD4. Now you tell me, CD8 are the cytotoxic cells and CD4 are the helper T cells. Helper T cells can stimulate both B cells to form antibodies or the cytotoxic cells. Okay. MHC class 1 is expressed by all nucleated cells of the human body. Understood? So, the natural killer cell, if it fails to recognize the MHC class 1, it will kill the cell. Then what is the role of the MHC class 2? These are present on the antigen presenting cell. Because it is present on the antigen presenting cell, suppose there is a dendritic cell, it will present both the antigen and MHC class 2, and that MHC class 2 will bind with the CD4, that is the T helper cell. Then the T helper cell will start processing that specific antigen, either to trigger the cytotoxic effect or the immunoglobulin production. Have you understood? There is one exception that you should remember. When I said MHC class 1 is shown by all nucleated cells. There are two exceptions. First is red blood corpuscles. Red blood corpuscles are not nucleated cells. Hence, during its lifespan, it will not produce MHC class. That is the reason why during blood transfusion, we are not concerned about HLA mismatch. We are only concerned about the ABO mismatch. Understood? The other exception is platelets. Why? Platelets are formed from the part of the cytoplasm and the part of the megakaryocytes. The megakaryocytes will have nucleus, and these molecules are present on the surface epithelium. Sorry. Okay. Furthermore, these MHC molecule or the HLA molecules are also associated with many autoimmune diseases. For example, ankylosing spondylitis associated with changes in the HLA B27 gene. Okay, so there are many like diseases. The last part of the lecture is the soluble products of the lymphoid cells. Now, all these things is happening. There is a definitive communication between the cells in the gland, the cells in the tissues, and the cells in circulation. This communication is mediated by some soluble chemicals which are again classified as lymphokines when produced by lymphocytes, monokines when produced by monocytes or macrophages, interleukins produced by WBCs, and chemokines which will lead to movement of various cells from the vessels or into the vessels. All those are collectively called as cytokines. These cytokines will have predominantly three effects. The first effect is called as the autocrine effect. Autocrine means it will produce the cytokine and the same cytokine will have effect on itself. It may have a paracrine effect. That means the cytokine will induce maturation, transformation or activation of a nearby cell, or it can have endocrine action. A very good example would be interleukin 1. Suppose you get an injury on your foot, it becomes infected. After the infection, what will happen? In the local region, there will be rising of the temperature, inflammation, but you will also get fever. Why? Because interleukin 1 will be produced in the local site, that interleukin 1 will come into the systemic circulation and will trigger the hypothalamus to raise the body's temperature. So those are called as the endocrine effect. These effects can also be differentiated into two types. One is called as the pleiotrophy. Pleiotrophy means suppose this one cell produces a one particular cytokine, interleukin 4 over here. This one cytokine will have many effect on very different cells. This is called as pleiotrophy. Similarly, the other thing is called as redundancy. That means many cytokines are produced, they all will have a singular effect on a particular cell. That is called redundancy. So both can be seen. Similarly, we can find synergism and antagonism. Synergism means suppose there is one cytokine, it enables to produce immunoglobulins. When it exposed to another cytokine, the immunoglobulin production increases rapidly. That is called as a synergistic effect. Antagonism means one cytokine will stimulate, the other one will inhibit. And last is what we call as cascade effect. Now, what does the word cascade mean? It will form a chain reaction or a feedback. That is positive feedback. That means a reaction that will go on and on. These are the various cytokines. When you will study deep, you will remember the most important parts. Interleukins 1, 2, 3, 4, 5, 6, 7. There are some interferons, alpha, beta, gamma, and tumor necrosis factor, TNF alpha, TNF beta, and some stimulating factors like GM-CSF, M-CSF, and G-CSF for maturation of the cells. Now, I already told you about the cascade reaction, the positive and the negative feedback. These can be seen in many diseases like septic shock or toxic shock syndrome, where the infection happens in a particular area, but because of the release of the cytokine, the various manifestations are seen. And if this is not controlled, it can lead to what we call as cytokine storm, and almost all organs of the body may be affected, and it may lead to many other symptoms like what we call as the macrophage activation syndrome or MAS syndrome. Sometimes it may even lead to HLH or hemophagocytosis. Okay, those are for different time. However, these cytokines are also used for treatment. For example, interferons are used for treatment of diseases like hepatitis, some of the malignancies, and can also be used along with conjugates to prevent many kind of tumors also. So not necessarily cytokines are bad, or it is uncontrolled. Some can also be used for treatment of many diseases. So there we come to the end of the lecture. I think we have understood about the immune system organs, the cells that are present in these organs, how they communicate with each other, and how they can action.